Good afternoon, everybody. Let's talk about sexual dimorphism. Sexual dimorphism claims in non-avian dinosaurs are underrepresented compared to other fossil groups, as you can see in the last 10 years of SVP annual meetings. Some attribute this to biology, saying that if you don't have dimorphism, then you don't have sexual selection, and therefore dinosaur ornaments were for species recognition only. However, this contradicts modern sexual selection theory, and species recognition is an integral part of mate selection ultimately. Others have suggested that male and female dinosaurs were both selecting for the same trait, resulting in no dimorphism. However, this is a relatively rare, steady state for a sexual system to be in, and it also requires in vivo experiments to determine why females might have ornaments. More other people have suggested that if you don't have significant bimodality, that you don't have evidence of dimorphism in dinosaurs. However, these are very low-powered univariate tests, and many sexually dimorphic traits are not bimodal. So what it does is it sets up a very high standard for the evidence you require. So for example, human height is not bimodal. We simulated human height by fixing female average height at 162 centimeters. And in order to get significant bimodality at something like a fossil sample size, average height for men needs to be 2 meters, which is absurd. And in fact, given what we know about the underlying population distributions, any significant bimodality at a more reasonable magnitude of dimorphism would actually represent a false positive. Some people recently have simulated growth curves for alligator and rhea, and they found that the rhea gives more significance when you apply t-tests. They attributed this to its rapid growth and suggested that because dinosaurs grew relatively more slowly, that's why it's difficult to detect dimorphism. However, again, this is a univariate test, and worryingly, the alligator data actually shows more prominent dimorphism with a larger effect size relative to low intra-sex variation. And actually, the results they're getting are a result of combinations between growth rate and lifespan, which makes it more likely that you sample at the region of the curve in which dimorphism is maximized. So what is the actual null hypothesis? Well, according to game theory, if you have unequal initial investment, you end up with different optimal strategies. So if you have a nesogamy, you predict sexual dimorphism. Statistically speaking, if you have finite male and female populations, you also might expect their means to differ by some amount. Now, dinosaurs were almost certainly sexually dimorphic. We can infer that they were nesogamic. They were very diverse, they were very successful, and they had elaborate ornamentation. For example, some of our largest ceratopsian specimens have horn tips that curve back down towards the skull, and this is very, very similar to the sexual variation you see in bovid horns. So when you have fossil data, you need to try to maximize your sample size, and you need to collect appropriate bivariate data. So look for features that are very commonly sexual. Look at ornaments, armaments, body size. But what you also need to do is get some sort of age proxy. So this could be lines of arrested growth, or it could be a body size proxy. Once you do that, you need to roll out the alternate hypotheses. The first one being non-sexual individual variation. So what you do is you identify a signal that's consistent with divergence in a growth curve. You then estimate the sex, and then finally, you constrain the maximum likely dimorphism and the confidence in your sex-based models. And of course, here, if you have less than 3 morphs, that can rule out many types of polymorphisms as well. So you look at your data and you try to see if you can find any spread or separation that might indicate this divergence in your growth curve as a result of dimorphism. So to do this, we looked at, we simulated dimorphic and monomorphic alligator and rhea data, kind of as before. And you can see that the dimorphic alligator splits very nicely compared to the monomorphic, but that actually the dimorphic rhea shows spread without separation. And it very worryingly looks similar to the monomorphic curve. Main takeaway is as you decrease sample size, the signals weaken. The next thing you do is you fit a curve to the whole data set and you look at the residuals. Do the residuals separate? Do they show any sort of delayed increase that might indicate this divergence in the growth curve? So if you look at the alligator data, the dimorphic data shows some nice separation. Also interestingly is that in the younger samples, you appear to have a difference between a long fuse and a short fuse relative to your maximum residual values in the whole curve. Now the dimorphic RIA data has this delayed increase in residuals. However, the monomorphic does as well, which means that this pattern can be a result of divergence as well as just a simple change in the rate of growth. So the next thing you do is estimate sex. You can do this in a variety of ways. I'm only going to talk about the simplest way, which is you fit a curve to the data and you assign sex either by positive or negative residuals. Now, since we're simulating this, we can measure the accuracy of these assignments, and you do usually get fairly accurate assignments. The pros of this is it becomes very easy to fit curves to these sexes, but it does assume some dimorphism in the juveniles, and it's also kind of an arbitrary division about the mean. But it is simple, so I'm going to use it as my example here. The next thing you do is you look to confidence intervals, prediction intervals, and AIC in order to constrain your maximum effect size, your maximum dimorphism, and also to help determine the confidence you have in your sex-based models, rather than significance testing. So you can see that R at the lower sample sizes doesn't do very well at fitting curves to the relatively more complex Gompertz equations. But the main takeaway here is that the alligator data performs very, very well. So you see that even at very small sample sizes, you get PI and CI separation. But in the monomorphic data, you do not get PI separation even at very, very high sample sizes. The RIA performs very poorly. Even in the dimorphic data at high sample sizes, you do not get PI separation. Oh, and the most important takeaway, as you decrease the sample size, intervals widen. So you can take your various regressions and you can model it either after a single curve in which size is independent or it's a function of age, or you can model it as a curve for each sex where size is a function of sex, or it's a function of sex and age. So what we see is in this example, the sex models outperform the no-sex models, and this is probably due to the way we estimated sex. We do it in a very logical way. But the main takeaway is actually that the relative preference decreases with decreasing sample size. So again, alligator performs really well. The dimorphic alligator shows a stronger preference for the sex-based model than the monomorphic data. However, the RIA shows similar preference for the sex-based models, whether it's dimorphic or monomorphic generated data. And you might have noticed that the RIA curve does not really perform too well on this method, and that is because the asymptote brings us back to the original problem of univariate statistics. It's not a growth curve. There's no growth at the asymptote. So contrary to previous claims, I think we should actually avoid asymptotic growth, particularly when the growth rate is fast and you have a long lifespan, which means you have more time at the asymptote. So dinosaurs didn't grow as rapidly, most likely, but their body size did asymptote. So I would say instead we should look towards ornaments. Do we have the opposite problem here? So univariate significance tests are low power and tend to give us false negatives, whereas our method is high power and can give false positives. However, with fossils, you expect low sample size, and as sample size decreases, power decreases. So the Stegosaurus mucide data from my 2015 paper has some of the best support within these dinosaur data sets. You can see when you essentially have size and shape axes, you get strong separation of the data and the residuals. If you use my morph assignments from my 2015 paper, which can constrain the assignments from plates from the same individual, you see strong interval separation and support for the sex-based models. The other alternative hypotheses include ontogenetic variation. We already controlled for this a little bit. But you can go further and see if you can find growth cessation in both morphs. You can rule out interspecific variation by finding coexistence of the two morphs without evidence of niche partitioning, such as a bone bed where the only thing that appears to be dimorphic is your ornaments. You can rule out taphonomic variation by controlling for completeness and plastic deformation. And then there's also case-specific hypotheses, such as inter-individual variation, So for example, it's been claimed that bimodality in PC2 of the stegosaur data suggests inter-individual variation. However, what you see is if you compare it to articulated specimens of other species of stegosaurs, the underlying angle distribution driving this is bimodal in itself, so it suggests we have a complete plate series. And then finally, if you believe you have high support for sexual dimorphism, you can then begin to question some of the biological implications, such as non-mating social behavior. And I believe I'm close to time, so I'll wrap it up. So thank you to my collaborators.